Good evening, participants. Am I audible to you? I think I am audible to you. I checked my, yeah. Okay, now, good, thank you. Now, let me take you on today's session on BLAST algorithm, which is left on yesterday. So I'll be teaching you how the BLAST algorithm works in virtually. And we come across what is the difference between PHI-BLAST and PSI-BLAST, and how this varies, and how about the algorithm, how we have to work with these tools. And we also discuss about FASTA and how to distinguish the BLAST and FASTA. Also, we didn't see practicing Needleman Once and Smith Waterman through software tools, isn't it? We just practiced the algorithm alone, like how to solve, like how to get alignment score and how to get a best alignment from the sequences provided, isn't it? So today we also see hands-on experience with the tools for in both the Needleman ones and Smith Waterman algorithm. So these are the today's session contents for day 8. Okay, this is last session for sequence analysis. From tomorrow onwards, we discuss about phylogenetic analysis. Okay, that is on day 9. Okay, now before going to today's session contents, can we recap the last session contents? Yeah, so we discuss what are the last session topics? What are the major topics we have discussed? Or can I ask you questions? If I am comparing human and yeast, okay, what amino acid scoring matrix method can be used? The question is, if I am comparing protein sequence of human and yeast, what amino acid scoring matrix method you choose? Yeah, participants, come for interaction. Yesterday you interacted well compared to today. Try it out. Right or wrong? Pinky, pinky, ponky. That's it. TLC, ah. What happened? What happened with TLC? How come thin layer chromatography comes into play? Ah, Kirtana answered blossom. And also another Kirtana answered blossom. Shreyasi blossom. Blossom is the exact answer. Okay, so your answers are right. Blossom is what we use. You know, if you go for distant related species, we have to choose this matrix, this method. And we also seen the blast, isn't it? So tell me the rank, how we rank the alignment in blast. How to rank the sequence alignment in blast tool. So if you have a query sequence, and if you are using a BLAST tool to search for a related sequence in a database, then whatever sequence you get for comparison is the database sequence. You have a query sequence and database sequence compared as pairwise sequence alignment, isn't it? So if you go for pairwise sequence alignment, what are the various scores you generate from BLAST? What are the various ranking scores? What are and also the second question is I have to ask about the outputs of BLAST. Before going to the outputs, can someone tell me how to rank the output of, in BLAST tool? Very good. Your answer is right. E value. This is the most important. And what are the other scores, just like E value? Bit score, good. E-value, bit score, and what else? That's it. Alignment length. No, actually, okay, this is fine. This is one of the parameter. You're right. Query coverage. Good. That is query coverage. Good. Query coverage you represent in percentage. E-value in form of float value. Okay, in decimals you get. And bit score, the form of positive score. So bit score, if you get, you also get raw score, isn't it? So bit score, raw score, other we get like max score, huh? We get like max score. Okay, can we see how we can, sir, and okay, I show you and then I can go for the next question first. Blast tool, you can see the output. I just shown two, no, but you get scores like yeah, max score, bit score, e-value, query coverage. Okay, this is the most significant you can remember. Other than this, you can also find the alignment, sequence alignment. If you click the sequence alignment, you can see the pairwise comparisons. Okay, no need to waste the time. Go for the outputs of BLAST. What are the various output formats of BLAST tool? So this is how we can anticipate questions in common entrance examination like GATE and BINC, Bioinformatics National Certification Exam. They give various examples in multiple choices, and they give something odd. You should correctly able to pick up the odd, even if you know all the information, all the output formats of the class tool. So one should try this. Okay, if only if you try this and use it for practically, then only you can. FASTA, Blastex, no. Paradise is one. Oh, see, everyone is uh, not sure on the answers. It's not confident on the answers. You remember ASN point one, XML, text. I think even faster is also a kind of an output, it seems. Okay. Yes, good. XML. So all these things, XML, we have two versions of XML as an outputs. Tabular, good. So these are some of the tabular is not a file name, but you can also find in a tabular format, isn't it? So if we ask about the we should also be confident on that. So one should try that. Okay. Today also we'll be trying with PHI-BLAST, PSI-BLAST. So we explore once again what kind of outputs and how to download all this. A very good JSON, JSON. Okay, that is also one of the output. You're right. Okay, I think few participants are very interactive. Okay, for providing even the correct answers from the beginning. Even I anticipate from the others. Okay, so let us practice. Okay, on today, if not practice for the past 4 days. Okay, because today I'll be completing the sequence analysis. Okay, you remember participants? Yesterday I was showing you something about MEME. Okay, so I also given a job name and my email ID to fetch the results. If it takes more time, okay, even through the email, we can receive the results. So we got a job, okay, worked successfully. But these jobs will be saved only for a week. Okay, if you want to save it, you can better save it as PDF format and you can keep it in your system. Okay, so whatever generated will be in temporary memory for in the server for a week. Now I'm going to show you that multiple EM for motive illustration. This is the tool used for multiple motives. If you remember on yesterday, I have taken five different species of insulin protein, insulin ligand protein, and I compared, and that I given as an FASTA format as an input, and I got a motif, okay, collective motif for all the five sequences. For this, how we can see the output? How? Oh, only three. I got only three slot because requested number of motifs three found. Only three motifs are found. Actually, if you see here, this is how we can find. Okay, this is the output you can find here. So human you can find here. So we are observe here. So you find the sequences are aligned properly in green, but in red, this red alone we have to adjust. You see, if you can give some one or two gaps, the red will be moved like that. Okay, then obviously you can find more pattern matching, isn't it? I better use the pen. Yeah. Okay. If I keep the red, you get it. Okay, and similarly here, see, for human, if you want to align it, again you have to introduce few gaps. And even here you have to introduce few gaps. You have to introduce more gaps. And these are already aligned. Human and CAVP. So already aligned. So you understand. So if you want to make maximum alignment score, so you have to use a local alignment, and you can adjust according to the motive sequences. Okay, and you can show this is the best sequence alignment you got from the local alignment. Okay, of course you are going to use default Smith-Waterman algorithm. Even you can download this output as PDF format or in the form of picture format in the form of SVG. Okay, and you can also find the frequency of amino acids in this 110 amino acid protein. Okay, and about the motive with some informations are shown here. So this is the major output you got for multiple motives. If this is for this is the software for finding the motive for multiple sequences. On yesterday, I also provided demo to get motive for a single sequence. You remember how to find a motive for a single sequence? What is the software I showed you on yesterday? I showed you a software to get a motive for a single sequence. For obtaining a motive in a single sequence. Can can participants can yeah, provide me the answer? Very good. So before typing the question, I got the answer again from Shreyas. Scan prosite is the exact one. Okay. So scan prosite is the tool. It is present in which web server? XPASI server. The scan prosite is a tool present in XPASI web server. Okay. Good. So your answer is once again right. Now I also showed you how the MEME results looks like. Okay. Now let me take you to the continuation of what left out on yesterday. You remember on yesterday I showed you some kind of an output in BLAST, and I also explaining you something about E value and bit score. Okay, we'll be discussing about that also at present. And before that, we can see BLAST algorithm. So what is how the BLAST is going to give us the output? Actually, majorly we have three steps in BLAST algorithm. The first step is the localization of the hits. This is the first step. According to this, the database sequence D is scanned for all hits T. So if this is your query sequence, the D is your query sequence, it searches for that database sequence, but you take only one sequence at a time. Okay. So whatever you take T of W match S in the list, and the position of the hit is same. So initially go for localization of the hits, and the second step is go for detection of hits. Hits, I mean the best database sequence what you get from the top to bottom. You know that the best hit will be present in the top. Okay, if you paste a sequence or if you paste the identifier, okay, if it is an NCBI, you have to type a GenBank accession number, or you have to type GenInfo ID. If it is an Uniprot, you have to type Uniprot ID, or you have to type the sequence FASTA format of the sequence. Okay, whatever you make it out. Okay, so, in all the softwares, you get the first thing is what the base hit. Okay, so first you have to localize the hits by comparison, and you have to detect the hits. So for this, all pairs of hits are such that have a distance of at most a. Think of them lying on the same diagonal in the matrix of Smith-Waterman algorithm. So you're going to find, you know that if you put a kind of a matrix, okay, so even in dot matrix method, in the diagonal, you can find more number of matches. So you can find is a line, okay, a continuation of the line. Okay, similarly, here even in this BLAST, okay, wherever you can find matches, especially find more matches in the diagonal side. So obviously you can find them even if you go for a trace back. Okay, the best alignment starts in the diagonal. Okay, so the defaultly the best it will have a good kind of trace back in the diagonal. And extension of high segment paths. That is, each such seed, that is S comma T, is extended in both directions. Actually, in BLAST, if you have 110 amino acid protein of insulin, and if you search it in any databases, okay, so whatever it's going to give us as an output, okay, we are not going to take the entire sequence, but we break into small pieces. So for the blast, it's generally going to take is not going to take an entire sequence for pattern matching. So instead of that, it breaks the large sequence into segments, which we call that as a high scoring segment pass. Okay, we call that as, so you can find somewhere. Where is that? Yeah, high scoring segment patch. So it breaks into multiple segments, and wherever you find high scoring in the segments, okay. So through the segments, if you join, obviously you get a more steep line in the diagonal side. Okay, so this is the extension of high segment patch is the third step for blast. Okay, the first step. The first step is the localization. Second step is detection of it. The third is the extension of high segment patch. So these are the steps of BLAST algorithm. As I already mentioned, if you execute an output, you get for the maximum alignment score, okay, you get a kind of red color, and next to that pink color, green color, blue color, black color. So in this output, you get only the pink color. Why we are not getting this red color? So if your sequence or query sequence is not matching with the merge characters in the database sequences, okay, and also that is one of the factor. If your query sequence, okay, is not matching, okay, much with the database sequence, you get this kind of pink color, or sometimes even you can start with a green color. So you can find from top to bottom, you can find the colors will be from red to black color. Okay, so this is one of the factor. And the second factor is if your query sequence length is very less. If query sequence length is very less, okay, then obviously you can also find it starts from the pink, or even it starts with the green, because based on the number of characters in the sequence, the alignment score increases. So the first factor is based on how much matches you have if you compare query and a database. And second is, well, what is the total size of the query sequence? If the characters are very less, like a 50 or 60 characters, then obviously it starts from pink or green. Okay, even if you get a better match in the database sequence. Okay, so this is something about how you can find in the graphical display. One few results are shown here, bit score and e-value. In BLAST, there is also we can also measure the significance of BLAST through e-values. The significance of BLAST, okay, is generally measured through e-values. Even this is an important question. Okay, we can anticipate. Can anticipate in uh, GAT, the Common Entrance Examination. The significance we can able to rank as highly significant, highly significant, and significant, and less significant. Okay, like how we give like a good, average, bad, something like that. So we can able to rank the sequence alignment through and which we call it as the significance of BLAST is measured through e values. If the e value Okay, if E value is less than e power minus 6, okay, then it is highly significant. If e value is in between e power minus 6 and e power minus 3, it is significant. If e value is greater than e power minus 3, then it is then it is uh, less significant. Okay. So for blast, we measure through e values. 1 minute. Actually, the score, whatever I mentioned, uh, is wrong. Actually, the more significant, that is highly significant, p value minus yes, yes, okay. E power that should be less than e power minus 6. That's what I mentioned, isn't it? And significant e power minus 3 to e power minus 6. And e power, if so, this is how we can rank the highly significant, significant, and less significant. Okay, so in this case, you can find whatever value got is less than, not greater. Sorry. In this case, you find it is less than e power minus 6. So it is highly significant. Not only this, all these values, whatever shown is highly significant. Okay. Uh, so just confirming. It's actually I mentioned like the lower the value, the better the acceptable value. So different books uses different thing. I am checking which is the common thing they're using. Okay. Actually, we can find like this. Actually, this is right. Actually, whatever they mentioned is right. E power minus 5. Okay. So I was explaining about e power minus 6, isn't it? So only for FASTA, even in FASTA, we have e value. So whatever I described earlier, e power minus 6 below is highly significant. That is for FASTA e value. And e power minus 3 to e power minus 6 significant for FASTA e value. And if it is greater than e power minus 3, then it is less significant in FASTA. How about in BLAST? In BLAST, okay, the e value should be e power minus 10. If it is less than e power minus 10, it is highly significant. And if it is significant, okay, the e value, if you give like capital E, okay, that should be in between e power minus 10 and e power minus 5. Okay, and if your expectance value, that's what we call it as the e value, if it is greater than e power minus 5, then it is less significant. So whatever earlier I demonstrated is only for FASTA. For FASTA, the value is less. That you have to remember. and for BLAST, the value is more. So the range for FASTA is e power minus 3 to e power minus 6. But whereas here in BLAST, we have e power minus 5 to e power minus 10. Okay. So if you compare these two, then you won't forget. Okay. So don't forget, this is for highly significant. This is for significant. This is for less significant. For BLAST, earlier whatever I explained you is for the FASTA tool. Even in FASTA, you get the expectancy value. E meant for expectant value. Okay. So no doubt in that. Okay. You can also check from other sources. You get this as the common one. Now let me take you to the formula for E value. Okay. So for the we also discussed this on yesterday. Like K and lambda or probability functions. That is background probabilities of the symbols. And this is the query sequence length. And this is the database sequence length. Okay, you understand? From the length of query and database sequence, the E value is calculated. If the sequence length is less, also you get a E value like that. Okay, so the E value is corresponding even to the sequence length. Okay, and the bit score is, of course, is also dependent on the alignment score, the raw score, which we use it in the calculation. Okay, and now we can discuss about the PSI-BLAST. What is PSI-BLAST and how the PSI-BLAST is going to work? So before going to the uh, illustration of the flow chart, okay, let me just give you some common understanding of what is PSI-BLAST and what is PHI-BLAST. PSI-BLAST is position-specific iterated blast, and PHI is pattern hit initiated blast. So you already know the expansion of blast tool, basic local alignment search tool, basic local alignment. You got it? So you get a maximum alignment score. Try to get a maximum alignment score by adjusting even the conserved regions during the sequence alignment. Not only that, we are also using the substitution. We are including the amino acid substitution scoring matrices, either PAM or BLOSUM. Okay, so this is how we can make it. And also yesterday, you remember, I told you some correlations of PAM and BLOSUM. Okay, I think I have to ask few questions because there is a lot to remember. If only if you remember, then only I can go ahead with this other concepts of blast. Okay, my next question is, um, uh, what is the equivalent value of PAM1? Can someone tell me? I mean, what is the BLOSUM equivalent value of PAM1? What is the BLOSUM equivalent value of PAM1? BLOSUM we have some range, and also uh, the PAM we also have a range, isn't it? So what is BLOSUM? Blocks substitution matrices. PAM percent accepted mutation. Can someone tell me what is a BLOSUM equivalent value of PAM? One. So I think before asking you this question, can I ask you the range? What is the range for PAM and BLOSUM? First, you can try this. What is the range for PAM and BLOSUM? Then we can go for the second answer. Yeah, try to answer quickly because I have to show illustration with the tools. FASTA tool, PSI-BLAST tool. I have to go with the needle and water. Yeah, even you can search like that. Just now I searched in Wikipedia, I mean, in the Google, isn't it? You didn't get an answer. What is the range? Yesterday I explained you the PAM. You have range from 1 to 250. Very good. You tried well. So PAM, if it is 1 to 250, BLOSUM, the maximum range you have like 10 to 100. So this is the range, isn't it? So if it is PAM 1, So you're mentioning PAM 250 is Blossom 30. Is it 30? Are you sure? Can we go to the slide? Good, right, well. So just because of Akshita Reddy, I'm going to show the PowerPoint, or else I might have waited for some more time. Okay, now let me take you to the slide, PAM and Blossom. Yeah, you should not forget now. So see this. So PAM 250 equivalent here is Blossom 45, and PAM 1 equivalent is Blossom 80. Isha's answer right for PAM 1. But for PAM 250, it is Blossom 45. And for PAM 120, which is used defaultly, that is Blossom 62. Blossom 62 is what default used in Blast tool, which we can find in advanced options. Okay, so these are also very important questions in common entrance examination. Okay, at least remember these three values, PAM 1, PAM 120, and PAM 250. Okay, yeah, so now let me come to the illustration of PSI blast. Okay, so if you see this PSI blast, position specific iterated blast, okay, I take you to the, before, I take you to the blast tool, NCBA. I take you to the NCBA, get inside the NCBA, take a sequence. If you already have a sequence, you can directly go to the blast tool. I think we already have a sequence on yesterday downloaded, isn't it? So I'm going to the blast. If I want to try with the protein blast, if I have a protein sequences, I'm getting inside. So PSI blast, PHI blast are present here in the bottom. Okay, so PSI blast, position specific iterated blast. Here only the position is most important. Okay, instead of pattern. Pattern example is if you have hemoglobin protein, if you have globin proteins, and if you're taking a proteins like hemoglobin, myoglobin, like hemoglobin, and other kind of globin proteins, beta globin. So the pattern here is a globin fold. You know, the structures of all the proteins, whatever I mentioned, can be hemoglobin, myoglobin, beta globin. The structure is almost similar, most similar, but the sequence alone you find variation in characters. That is, you find more mismatches. Okay, so pattern is same, but sequence, the matches in the sequence are very less. Okay, so if you find, or if you want to focus on patterns, then you focus on patterns, and if you want to focus on positions, focus on position. Okay, now this is position specific iterated plan. You have to either give accession number, or you have to give the FASTA sequence format. Okay, so I just try with the FASTA, but I have the FASTA format from Uniprot, isn't it? Let us try with that also. I take one protein, insulin protein. Okay, it's opening in the meta. I like to open it in Notepad. Yes. So just opening this sequence. Okay. Better I take human. I take a human insulin protein, but you have an Uniprot ID. I think try. So I just pasted this sequence. Okay. And I'm going to try with the PSI blast. Default, I keep it in NR. Okay. And if you're confident about the positions, you give the positions. Okay. For example, if I want to give position like 97 to 105, okay, only these positions alone are taken in major consideration. Okay, so if you have 110 amino acid of a protein, but this is something more significant to search, I can also give a kind of a position specific. Okay, and I can click show results in new window. Before that, I can also show you some algorithm parameters. You have algorithm parameter whether you want to get 500 outputs, or you want to get less outputs. Okay, whatever you want, you feel you can select that. And threshold, word size. Okay, so that is actually I told you in Blast. So not the entire sequence you take for comparison. You break that into segment pairs, and whatever high segment pairs you use, you just assemble it to get a kind of a good kind of alignment, sequence alignment, isn't it? You join all the high segment pairs. So here, the word size for the protein, you default I take it as 3. Okay, for nucleotide, it varies. Nucleotide, it is 6. Okay, so just because I'm using proteins, the default size it has fixed is 3. And for block substitution matrices, Blossom 62. Okay, Blossom 62 is the default one. I told you the equivalent value is PAM 120. So either you can keep Blossom 62. If you have PAM 120, you can keep PAM 120. Okay, everything works similar. Okay, no doubt in that. And the gap cost, the default, we have all these parameters. Now I just want to see this in the new window. I'm not varying any other parameters except the target sequence alone. I just varied as 250. Simply check the box and click the blast, so you get a kind of a thing. Okay. Still, I used only the Unicode FASTA sequence. We are getting an output. So no doubt in that. So one can even download the sequence from the Unicode. If you have a problem to find exact sequence in the NCBA, and then you can go for a blast tool, application of PSI-BLAST tool in the NCBA. Okay, so we'll be getting an output shortly from the PSI-BLAST. Before that, I can also show you an output from another software tool called Emboss Needle. You know, there is a package called Emboss, that is European Molecular Biology Open Software. So here I told you 151 programs are there, and one such program is Needle. This program uses the global sequence alignment. Okay, if you simply go to this Emboss Needle, and if you want to try the pairwise sequence alignment of two sequences, paste the two sequences as an input, and then you can click submit. Okay, you will be getting output instantly, no doubt in that. But you have to paste the two sequences. Either you have to paste or you have to upload two different sequence in FASTA format. Okay, I already have a sequence downloaded, so I just pasting human insulin initially. And if you want to try with the pig insulin, okay, so scrofa. So I just copying it and pasting it in the second text area. And now, if you want to give a job name, you can give it, or simply I can click submit. Okay, so you'll be getting a result immediately. Now, if you want to view the result, click view results. So this is the result. Okay, I just zoom in and show it to you. So what is the exact output you can get from here? So you can get an output like the identities. Okay, you get output like identities, similarities, and gaps. Okay, identity, similarity, and gaps are the outputs of this emboss needle. So these are the default input parameters we already fed: gap penalty 10, gap extend penalty, etc. And you can also see the sequence alignment. So wherever you find mismatches, you find dots. Wherever you find matches, you find these vertical lines. Okay, so what do you understand from this? Out of 110 amino acid, so 94 matches are there, and some mismatches are also. This is not a positive, but this is a similarity. Okay, so positives is what? The number of matches and mismatches. So similarity is something different. Okay, so this is an output of embossed needle. However, we find an output for an embossed needle, which we worked in uh, work for global alignment. We can also try the same using embossed water for local alignment. If you want to try for the local alignment, so that is working with the Smith-Waterman algorithm, you can try with the embossed water. So similarly, here also you paste the sequences. So now I'm going to paste initially the first sequence as human insulin protein in the window 1, and the pig insulin in the window 2. Okay. So after this, okay, I am simply going to click submit. See, before submitting it, you can also see in the top, okay, you have protein and DNA. If you're trying with the protein, you would select the protein. If you're trying with the nucleotide, you can try the nucleotide. So here also for the local alignment, you got similar kind of a results. Okay, it depends, okay, based on what kind of a sequences you are comparing. Okay, so for the global and local, obviously can find the concept regions are same, so only you're getting the same kind of an outputs, the identities, similarities and gaps are same. If concept regions are something different, okay, and if you need a kind of an optimal alignment, then obviously you can find a kind of score. Okay, the variation with respect to score. Here the score is 484.5. Did you notice the the global alignment score? I didn't notice. You can check it. We make it less than this, or we make it the equivalent score. But you won't get more score than local alignment. That is uh, that is for sure. Here you got like 484.5. Okay, you can also check with the global alignment. However, I tried initially. Okay, now let me go for illustration of PSI-BLAST. Okay, so you remember what I did? I just given an input of a human insulin, and I just selected PSI-BLAST instead of BLAST-P protocol. Instead of BLAST-P protocol, okay, you remember? Instead of BLAST-P, I used PSI-BLAST. Okay, and then I showed the results in new window. Okay, there are also other types of blast. Okay, now let me see how you can find in PSI-BLAST. Okay, in PSI-BLAST, okay, I just tried with human insulin, but I've specified the regions. I specified some regions. Okay, so based on that specific region, okay, you got some kind of a sequences, but most of the sequences initially you find are from the human. Homo sapiens. If you want to find the next immediate species, which is closely resembling, you got like Odo coelius. Okay, this is the one we got, which is more closely resembling to that particular sequence, particular important sequence of human insulin. And the next is Pipistrellus. One can check that what is the species and can check the information on that. Okay. But if my objective is to work on PSI-BLAST, okay, the PSI-BLAST is of course position specific, and also we are also that is not just a position specific blast, but this is position specific iterative blast. What do you mean by iteration? The iteration is like if you want to check the sequence alignment, pattern matching, okay, through some advanced filters, through some specific threshold values. For example, if I want to go for second alignment, second iteration, now we got like PSI-BLAST iteration one, isn't it? In iteration two, if I want to use specific filters, for example, here I got E values. What is the E value got? 4.7, 6.3, something like that you got, isn't it? You can find increase in values gradually. But if I want to see the E value from 4 to 10, suppose if you want to go for checking a sequence alignment for 4 to 10, okay, that's because I'm giving a small range, okay, a small specific position, you are getting these values. But if you're trying with maximum protein size, you get obviously a less E value, no doubt in that. Okay, so now we can see the E value if you want to find the values from 4 to 10. Okay, this I'm using it as a filter. Okay, and now I'm going to run, click run. Okay, now this is going to do the second iteration for us. Initially, we use the positions as the primary thing. Your search parameters are adjusted to search for a short input. Yeah, we're going to get an output soon. So initially, we have given some sequence length like 97 to 105. That is what we have given, isn't it? Later, we are just adjusting the E values to get a best iteration. You know, what is the purpose of going for the second iteration? So whatever protein sequence you are going to search in the database, okay, we are going for a specific part of a sequence, and that too you have to search with all the 300 million sequences. So if you have 300 million sequences in the database, and if you want to check with all the 300 million sequences, you may miss a few sequences just because the computational power is more, and you may get some kind of a bias if you try with many sequences with the sequence alignment algorithms. Therefore, it uses Smith-Waterman, so obviously you need a computational power, and even if you miss one or two, okay, this will be checked, okay, once again in second iteration with the filter whatever you have used. The filter I have used is e power. So you got e values, yeah. So based on the e values, you got again the second iteration, you got some test output. The first test what we got is again Odo coilus. Next pipistrellus. Okay, and even if you want to go for another kind of filter, you can give again you can vary the query coverage. If you know if you're not convinced with 97 to 105, if you want to try with another position like 90 to 105, you can give a query coverage and you can click run. Then it goes to the third iteration. Now whatever you got is for the iteration 2. If you click here, then it goes for the iteration. So what do you understand from this? This is position specific iterated blast. Unlike blast, okay, so this is like an advanced blast. We can check with respect to positions. Okay, so the pairwise alignment is done through the position. And the same, we also have another kind of a blast called PHI blast, pattern hit initiated blast. Here, we have to feed the pattern. Okay, so for finding a pattern, you can able to find the pattern from here, whatever is shown in MEM. You can see this pattern. Okay, so where you can find this pattern? I think this is an insulin receptor. This is an insulin receptor. So when do you find this much large? I think so. Just compare with the receptor, isn't it? Let me check. So if I want to click this, what is the pattern? If you have some specific amino acid, okay, you can give that specific amino acid. Okay, that is 15 to 57. Start is 50, this is 1 to 6. This is 15 to 57. There's 17 to 57. So, okay, in pattern, okay, you can also give like 17 to 57. So that's what you can find conserved regions, isn't it? So 17 to 57, you can keep it out, and you can go for blast. So if you find a pattern in the motif tool, you can feed the sequence, the particular sequence. Okay, here, okay, this is the pattern. Okay, whatever pattern you got, and if you search it, you may get these species like tannery, pig, bovine. You have to search what is the best one from the pattern hit initiated blast. Okay, we'll be getting the output shortly. Before getting this result, we can go for another tool, that is FASTA. So next we'll be seeing FASTA. For working with a FASTA, so one can try like this Virginia FASTA server. First, I can show you how to work with the FASTA server. Then I can give you some theoretical explanation. So if you type Virginia spacebar FASTA or UVA FASTA server, it takes you to the this website. Okay, Virginia University has developed this kind of a FASTA program. If you want to work with protein FASTA, you can click this tool. And if you want to work with nucleotide FASTA, you can work with it. Actually, for protein, you can try with FASTA, and even nucleotide, again, you can use the same FASTA. You know, in BLAST, we have types of BLAST like BLAST P, BLAST N, BLAST X, T-BLAST X, T-BLAST N, isn't it? Similarly, even in FASTA, we have types. We have types like if you try with the translated DNA, we can try with FAST-X and FAST-Y. Okay. And if you want to try with protein versus translated, we can try with T-FAST-X and T-FAST-Y. So one should also first check this tool, okay, and then you can try with the corresponding inputs and can get an output accordingly. So defaultly, if you want to work with the protein FASTA, you can click this tool. If you click this thing, you will be getting an input window like this. You have to simply paste a query sequence. Now I'm going to paste sucralpha insulin protein as an input sequence. So if you give it this, okay, you ensure that you have selected a protein, okay, and also check the database. Here, the database what we use is the protein information resource, PIR noted in default. But you can also check the other databases if you want. Like for example, if you want to try with 3D structures of a protein, select this PDB structures of NCP. So obviously, you are going to select MMDB in this case. Okay, so now I just want the sequences and I want even the structures. Then we better keep this protein information resource database. Okay, default thing, whatever is there, I'm not changing anything. We keep it as such, but see the scoring matrix kept in default in FASTA. In BLAST, it is Blossom 62, but here it is Blossom 50. Okay, it is Blossom 50, which is around 25% similarities. Okay, so you find something related to that. Okay, Blossom 50 is default, but if you want to go with the Blossom 62, go with this. Okay, whatever you want, you can select it. And now, okay, I just kept in default. I am not changing anything. Simply click search database. You know why we call this as a FASTA tool? This is fast algorithm. So defaultly, it uses the global alignment. Okay, and this tool uses a kind of a fast algorithm to fetch a kind of a pattern matching, a sequence as soon as possible. The algorithm varies in FASTA and BLAST. I told you in BLAST we have three major steps. Here, if you see in FASTA, we have five major steps in algorithm. Okay, so based on the algorithm developed here, you can find the output is little fast. Okay, now is click search database. We just also check the timing. For the blast, you got almost 10 to 15 seconds to get an output. But here, within 1 to 2 seconds, 2 to 3 seconds, we got an output. So for the pig insulin, the most similar insulin protein matching is from the rat. Okay, so how I am telling? So based on these scores, okay, based on the so here you can find the scores like OPT, bit score, E value. You remember I told you about the significance of E value in FASTA. If you want to rank the significance of FASTA, okay, I told you E power minus 6 and below. E power minus 6 and below are the highly significant. If you get above E power minus 3, then it is less significant. In our case, whatever hits we have, okay, from here to here, that is from here to here, okay, and I told you it is E power. Most significant is up to here. The most significant, that is up to E power minus 15 is most significant. Here, if you take like this, okay, this is 2.8 into 10 power minus 4, something like that. 2 power 8 into 10 power minus 4 comes under significant, not highly significant. So if you want to see the significant range, okay, the significant range, I think you can find from up to here. I'm not sure. Up to here is significant. And this sequences are less significant. Don't take these sequences, okay, because we find less significant. So the significance we can measure like this, and also you can find personal identity and personal similarities, like how you find in embossed needle and embossed water. So even these are the other outputs you can find. And if you want to find the alignment, sequence alignment, click align here. If you click the first align, okay, that is that which is showing more similarity, which is the best hit showed with the pig insulin. Okay, so you can find the pig insulin at the as a query, which is at the top, and the database sequence at the bottom. And you can find the scores like init 10, init 1, okay, OPT, Z score, bit score, E value. Okay, so these are the types of outputs we generate in the FASTA. Okay, now I show you the output of PHI-BLAST. So in PHI-BLAST, there is no such, this PSI. Where's the PHI? This is the PHI-BLAST. Oh, no. I think this is the PHI, what I tried. Okay, whatever I tried. Okay, so this is the output. So the first pattern hit for the human insulin, okay, for the query, the pattern 57 to what I have given 57. I've given 50 17 to 57. characters. Amino acid, 17th amino acid to 57 amino acid. You got gorilla is the best matching one. After gorilla, pongo, I think kind of orangutan, okay, and chimpanzee. So these are the best conserved region. You can find the insulin conserved regions in this species. Okay, this you got from pattern hit. For the position specific, you got a different species, isn't it? Yeah. So this is how you can check the pattern hit and position specific iterated class. Now let me explain about the FASTA algorithm. Yeah, if you see this FASTA algorithm, okay, I told you the FASTA, you can use URL Virginia FASTA server, or you can also use EBI FASTA server. Okay, either use this link or this link. Okay, and if you see the FASTA server, you get a kind of a home page like this. So select a appropriate kind of a FASTA tool from the types of FASTA tools and feed the FASTA format, okay, or give an identifier and click simply search database. You are default using a protein information resource database. And you remember the FASTA algorithm uses hashing hashing the code word or k tuple method to find the best sequence alignment. Okay, you remember I told you the pairwise alignment, the second method, Ford or K-tuple method. So that's what it is going to use. And in Wikipedia, you will be getting this kind of an flash image, like how the Smith-Waterman algorithm is used. Okay, to get a best output. Okay, to get a best sequence alignment. Okay, so whatever Smith-Waterman algorithm, okay, is defaultly deployed in FASTA tool. Okay, this FASTA algorithm majorly have four steps. Okay, we also have fifth step, of course, that is to rank the significance of the FASTA through eval. Okay, so that I just skipped. Okay, but major if you skip that fifth step, we have totally majorly four steps. So what are the major four steps in FASTA algorithm? Okay, if you see this, the first step, what we see here is the identify regions of similarity. So if you compare sequence A, okay, if you compare sequence A and sequence B, wherever you find matches, for example, if you have a sequence from top to bottom, and here you have like this, okay, in the form of columns, okay, wherever you have matches, you use the dots. And if you join the dots, you get these lines. Okay, like you are using a dot matrix method and you're just going to join the dots. So obviously get suppose if you get like this, okay, in the first step, that is what we call it as a finding runs of identical words. So defaultly, in BLAST, I told you the word length what we take is 3. But here, instead of word length, here we call this as a k tuple length. The k tuple, I mean, in the FASTA, we call this as a k tuple. And here you can find the k tuple length for the protein default is taken as 2. Okay. And for the nucleotides, we take it as 4 or 6. But whereas in BLAST, the length is maximum 3. And I think this is almost 10 for nucleotides. Okay. And what I'm in the BLAST, we call that as a segments. Here we call this as a words or k tuple. And now if you see this, we just take 10 base diagonal regions found based on the matches and mismatches. Okay, so generally you take the matches alone in consideration. Mismatches, you ignore it. And now if you see this second step, we rescore regions. Okay, we rescore regions and identify best initial regions. So this is the second step. Okay, this is the second step. So we are using a PAM matrix. I'm going to use a PAM matrix and keep only top scoring segments. So these are all eliminated. See, this is in light background. Here you had initially in dark background, but now these are all present in light background. So we are ignoring these regions. So that we are just considering this by using this PAM matrix. So if this is the second step, the third step is after all diagonal elements found, try to join the diagonals by adding gaps. So not only you are taking these elements, but you in the third step, you are eliminating these fragments. So I take a different color. I take a different color. So I try to eliminate these fragments now. Okay, I just consider the diagonal elements alone, and I'm going to join the segments using caps and eliminate other segments. Okay, so you got like this. This is the third step. And the fourth step is what? You use a dynamic programming to create an optimal alignment. So just like that, if you use a dynamic programming, okay, you join all these sequence and you get something like the more similar matches are present in the diagonal line. Okay, so these are the major four steps, and of course, fifth step is to after getting a score, have to rank the significance based on the e values. So how a fast output looks like, this is how a fast output looks like, and these are the types of FASTA based on what query and what database sequence you use. You have to select the types of FASTA. Okay, and if you see the comparison of FASTA and BLAST and overall, FASTA searches for all matching words of length k. So I told you, no. So in BLAST we use a larger thing, the high scoring words. For proteins it is 3, but here it is for 2, and 4 to 6 for nucleotides. But here you take up to the 11, up to 11 you can take. And for a you can use a FASTA tool for calculating statistical unrelated sequences. You get a fast output here, but precision is less. But in BLAST you get more precision, but it the time consuming. Okay, and also you can see this difference in steps of the algorithm. Okay, so I've completed our the BLAST and FASTA tool and even how the algorithm works. Any doubts, you can ask me now. Any doubts you have? So of course the BLAST, FASTA, FAM, Blossom, these are all the most important technical concepts to be remembered for common entrance examinations, especially the values, and especially even I told you in BLAST what is the sequence length, what is the segment length, and how about in FASTA, K-word or tuple length, even that you have to remember. And what are the default parameters we use in amino acid scoring matrices, and what are the databases we use in FASTA and BLAST. Okay, like a NR, PIR. Even if they're given multiple choices, you have to correctly select them. So one should see that also. Okay, and also in the BLAST tool, you can, after getting an output, you check the outputs there at the top. Okay, so you can to find how we can improve on the BLAST outputs. So all these things are important for common entrance examination preparation. So one can read all this. Okay. If there are no doubts, thank you very much for your interaction. Okay, and so let us join on tomorrow's session in phylogenetic analysis.